Hello, my name is Margarita Cañarte. My topic is about tumors with small RNAs derived from transfer RNA. Definition: Small RNAs derived from transfer RNAs are an abundant class of non-coding molecules whose biological functions are not yet well understood. It was recently demonstrated under in vitro conditions that the inhibition of a specific tRNA induces apoptosis in rapidly dividing cells. This tRNA binds to the mRNAs encoding ribosomal proteins to improve their translations. A decrease in the translation of these messengers decreases ribosomal RNA processing, resulting in a reduction in the number of functional ribosomal subunits. This data establishes a post-transcriptional mechanism that provides a potential new target for cancer treatment.
At one point, people believed that introns, the non-coding part of our DNA, were just junk DNA. We thought they had no purpose. We know now that this is far from the truth. Recent research has proved several other types of RNA exist besides the messenger RNA that codes for proteins. Small non-coding RNAs produced in the non-coding part of the DNA were found to be extremely important for gene regulation in animals and plants. MicroRNA or mRNA, small interfering RNA or siRNA, and Pee-wee RNA or piRNA are all types of small non-coding RNA. They have in common their short length and ability to silence genes. They differ, however, in how they function and how they're produced. In this video, we'll be discussing microRNA, short RNA sequences about 22 nucleotides in length. They are part of a large endogenous RNA pathway. Endogenous meaning it was already existing in the cell, formed in the nucleus like messenger RNA, and wasn't introduced by an outside source. The sequences that code for microRNA are found in introns. These non-coding regions of DNA mean that they aren't translated into protein. Their purpose, however, is to regulate the production of proteins. MicroRNA regulates gene expression through RNA interference or gene silencing. As we know from the central dogma, messenger RNA transcribed from DNA is translated to synthesize proteins. Only when DNA is transcribed into RNA will microRNA jump in to stop the final step of the central dogma and prevent the protein from being made entirely. MicroRNA are synthesized in two stages. First, by a protein called DROSA in the nucleus. Then they are exported into the cytoplasm and processed further by a protein called dicer to make the mature single-stranded form of 22 nucleotides. Then once an Argonaut protein, usually abbreviated AGO, is bound to the mature mRNA, it produces an RNA-induced silencing complex, also called RISC. And then it's ready to get to work. It can silence genes because each microRNA sequence is partially complementary, so it matches up almost perfectly with its target messenger RNA sequence. MicroRNA binds to the messenger RNA sequence of genes that it needs to turn off. Argonaut protein helps it by lining it up. Then the chaos begins. By binding to the target sequence, microRNA has essentially created a roadblock in the messenger RNA to prevent ribosomes from translating. Not to mention that the Argonaut protein can straight up cut the messenger RNA in two places, destroying it. So a microRNA can therefore repress translation or degrade messenger RNA entirely to stop it from making proteins. In the video on microRNA biogenesis, we'll discuss in much more detail how microRNA is made and this whole process. And that's a wrap. Thanks for watching.
Application in medicine. A recent study carried out focus on the analysis of a small RNA derived from transfer RNA. It plays a role in the regulation of the number of ribosomes, and the greater the number of ribosomes, the more potential for the improvement of protein synthesis, which it allows for rapid cell growth and proliferation. These results suggest that dsRNAs can help fine-tune the diagnostic and prognostic by differentiating malignant from benign tissues, one type of cancer from another, and perhaps distinguishing patients who will respond to therapy from those who will not.
Today's episode is made in partnership with PANC-1, an international network of world-class cancer research institutions looking to increase the survival rate for pancreatic cancer. If you're interested in learning more or are able to donate to their research, head over to panc1.org or click the link in the description. Around the turn of the 21st century, researchers finally found evidence for a hypothesis that had been around for a while. In addition to protecting us from diseases, our immune system helps to protect our body from itself by watching out for cancer. And that, of course, raises the question, could we give our immune systems a leg up in spotting tumors? Could we develop vaccines for cancer? The idea is fairly new, but there are already 2 cancer vaccines on the market, and researchers are looking to up that number with a technology that promises to be cheaper, faster, and more customizable than traditional approaches, vaccines based on RNA. Traditional vaccines expose our body to antigens, proteins or sugars derived from pathogens like viruses and bacteria, which our immune system can recognize, learn from, and use to attack a real invader. In cancer vaccines, those antigens come from tumor cells instead, but the idea is the same. We want to train our immune systems to find those antigens and clear out the tumor they were derived from. One approach is to use whole tumor cells taken from a patient and modified to serve as training wheels for their immune system. Others only use the antigens from the patient's tumors. But cells and bits of protein aren't our only options. Enter vaccines based on the genetic code, the nucleic acids, DNA, and RNA. This approach is meant to be faster and easier than traditional vaccine development. DNA and RNA vaccines reprogram cells to churn out that antigen without having to expose our immune systems to any external proteins or invaders. With DNA vaccines, you make a DNA molecule that carries instructions for the cell to make some antigen, using the same genetic code and protein-making machinery it already runs on. Then you stick it into cell's nucleus somehow. After that, the cells copy those instructions in the form of a piece of RNA, which travels outside the nucleus to where all the proteins are made, and finally, the cell starts pumping out antigens. Meanwhile, with an RNA vaccine, you just skip those first steps and put the instructions on the RNA molecule to begin with. The idea is that we can very easily put together nucleic acid molecules to code for whatever we like in the lab. Not so with proteins or whole tumor cells. So this approach is fast and it can be tailored to whatever we like. There aren't a huge number of these things kicking around yet. While some DNA vaccines are approved for veterinary use, more and more research focuses on RNA instead. And that's because it's harder to get the DNA where it needs to be, inside the cell's nucleus, where it's going to be turned into RNA and then sent back out to make the antigen we want. And once the DNA gets there, it can cause problems. Like sometimes the cell can incorporate loose DNA into its chromosomes, and it's not picky about where. Rarely, that can interrupt other genetic material you really need, and ironically, that's a step toward cancer.
Pros and cons. Pros are relevant to combat the onset and progression of tumors. Helps diagnostic and prognostic by differentiating malignant from benign tissues. Cons. The treatment is paid for by National Health Service, costs about $500,000 per patient per year, and is for life. The laboratories that develop the new therapies assure that developing them takes many years of investment and research, and that since they are rare diseases, the number of beneficiaries is low.
Number 10. RNA-based therapies.
A code of genetic information. Ribonucleic acid has become a popular target for therapeutics. Interfering with that code gives scientists the ability to intercept a patient's genetic abnormality before it is translated into non-functioning proteins. Antisense nucleotide therapy prevents the production of proteins encoded by a disease and is being explored in several conditions such as age-related retinitis, familial hypercholesterolemia, and Huntington's disease. RNA interference, a mechanism for gene silencing, is currently being explored for use in cancer and neurological diseases. With RNA interference, RNA molecules, either microRNA, miRNA, or short interfering RNA, siRNA, inhibit gene expression by neutralizing targeted mRNA sequences. FDA approved in 2018, the most successful RNA interference therapy is for the treatment of hereditary transthyretin-mediated amyloidosis. Feeding off the success of DNA-based gene therapy, RNA-based therapy puts a twist on treatment, sparking progress and innovation in therapeutics.
Issue update in Ecuador. In Ecuador, the subject of RNA therapies are not known by many. However, research has been carried out based on other scientists in which these techniques seek to directly edit or correct DNA within genes, but RNA therapy acts in a different way. Conclusions. Messenger RNA technology could be the way out to correct genetic problems. This in turn will make it possible to cure diseases such as cancer and even HIV. Vaccines with messenger RNA technology were listed by Time magazine as one of the innovations of 2020. For this reason, researchers around the world have begun to study this technology to treat chronic and catastrophic diseases such as cancer and HIV.